thank you, Courtney, for such a nice introduction. And myself, Nitin Kadam, I'm a PhD student at CSIR-National Institute of Oceanography in Goa. Soon I'll be defending my thesis. So today I'll be talking about the magnetofossils and the oxygen minimum zone in the northern Indian Ocean. Some of the talks today earlier had given us a little bit of idea about what are oxygen minimum zones and where these zones are in the different parts of the ocean. So the first part would be an introduction. I'll talk about the biomineralization, different types of biomineralization, then magnetotactic bacteria. There are different types of crystals that are produced by these MTBs and their habitat, their significance. In the later part, I'll be talking about the magnetofossil and how they are useful in reconstruction of paleo-oxygenation conditions. So there are two types of biomineralization. Basically, one is direct, that is biologically controlled, where we have a strong genetic control of the organism that produce these minerals. The example of such a type of biomineralization is the formation of foraminiferal tests. In this example, this is an example of a foraminiferal test of species Globigerina ruber, that is, used for fast oxygen conditions. And another is indirect precipitation, that is biologically induced, where we have indirect control like metabolic activities of an organism that helps in the formation of different minerals. The example of it is magnetite formation by the disseminatory iron reducing bacteria. And biomineralization actually plays an important role in biogeochemical cycles because they control the movement and storage of different elements such as carbon, calcium, phosphorus. So yeah, studying these the biomineralization is important for this reconstruction of these biogeochemical cycles. So magnetotactic bacteria, the mineralization by these MTBs is a great example of biologically controlled biomineralization. MTBs are a group of prokaryotes. Those crystallize iron minerals, specifically magnetite or greigite, usually in the size range of 20 to 120 nanometers. And this is an TM image of a species, Magnetospirillum griffinwoldenis, that's showing a distinctive cell of a bacteria and the magnetosome crystals. So EMTBs have a strong genetic control on the formation of the different crystals in their cell, and different proteins basically helps in the formation of these crystals from the first stage, that is from vesicle biogenesis up to the chain assembly and positioning. Some of the examples of these proteins are like MMM and MMH that helps in the for iron uptake and magnetite crystal nucleation, and others are like MMAK, MMJ. These proteins helps in chain assembly and positioning. So these crystals are usually aligned in chains, and so forming a magnetic dipole, and that helps a bacteria or that gives an upper hand for the MTBs compared to the other bacteria to find the optimal condition in Earth's magnetic field. So, and this process, the movement of these MTBs in Earth's magnetic field, this process is called as magnetotaxis. So, magnetotactic bacteria, MTBs, regulate the mineral composition. Usually, they form magnetite or greigite, and also there are different forms or different shapes of these crystals produced in these MTBs. For instance, this is a cubo-octahedral magnetite, octahedral magnetite, some bullet-shaped magnetite crystals. So yeah, so and also these MTBs regulate their crystallographic orientation within their cell. So where these MTBs live, what are their habitat? They usually prefer to live in a zone or called as oxic anoxic transition zone. Why? Because at this particular zone, uh, they can they can optimally draw maximum nutrients for the growth, and you will find these MTBs in great abundance as at these zones. Basically, a transition, oxic anoxic transition, it's that comes in a suboxic range, and usually magnetite producing MTBs occur at this zone, oxic anoxic transition, but in that greigite producing MTBs will occur down below to that zone in sulfidic environments. So the main point from this is that MTBs are sensitive to the oxygen concentrations in the water column. And as we know that MTBs, they sequester these elements like mostly iron in their cells. So depending on at what time scale these they hold this iron in their cell and they release into the system, so that will significantly affect the biogeochemical cycle of iron. Not only the iron but also, and importantly, carbon, nitrogen and sulfur, because most of the magnetite producing MTBs are nitrite producing. So what are magnetofossils? When these magnetotactic bacteria, the cell dies, these crystals, they become part of the sediments, and they basically they provide a signal, a magnetic signal with the bulk other detrital magnetic signal. And these crystals or the magnetofossils produced by MTBs are called conventional magnetofossils. In the later part, it will like this will give a clear idea why we call it as a conventional magnetofossils. So it has already, I mean, it's well established and in found in several studies that the environment where we have a less organic carbon and higher oxygen levels, you'll find magnetofossils with more of a equant shape or isometric shapes. And the conditions where there we have a more organic matter and lower oxygen levels, you'll find magnetofossils with elongated shapes. So this will, I mean, this is an important observation and it will come later in the presentation. So yeah, and we have another type of magnetofossil. We call it as giant magnetofossils. These magnetofossils are larger in size and morphologically different than conventional magnetofossils. They are the size range is on micron scale, and until today, the occurrences of giant magnetofossil, I mean, the giant magnetofossil occur consistently with the conventional magnetofossils. And since their discovery, first report is in Schumann's et al. 2008 paper, where they reported different shapes of the giant magnetofossils. Initially, I mean, four shapes of these giant magnetofossils are reported. First is a spearhead shape, another is bullet, spindle, and needle, and all these are of a magnetite composition. And recently, recent studies has proposed new shapes that are seeds, squash, spades, and more recently, king, giant bullet, and sock shape. Yeah, and all these are like yeah, all these are like magnetite composition. So so the current challenges in this the field of research is like the first I list here some questions like what is the origin of giant magnetofossil? It's still, I mean, the question needs to be answered, and I believe that answering this question would solve the whether these magnetofossils are really biogenic. And another is if they are biogenic, what is the function of these crystals inside if produced by so many other organisms? Another question is why are giant magnetofossils always co-occur with conventional magnetofossils? I mean, there are no records that shows that only the magnetic signals are dominated by only giant magnetofossil or conventional. Another thing that is the second part of this presentation is, what is the relation of between oxygen minimum zones and giant magnetofossil organisms, and also the conventional magnetofossils. And another question is, that is a challenge, how diagenesis, mostly the reductive diagenesis, affects the stability, composition, and detectability of magnetofossils across diverse depositional settings, such as, because the degree of diagenesis will be different at different environment. Let's say for the shelf regions, because of the more organic matter, one would expect that there will be more reducing conditions in your, in a sediment core, compared to the deeper water where we have a less organic carbon. And the last question or the challenge would be, how can the impact of this reductive or sulfidic diagenesis can be quantified, to accurately reconstruct the magnetofossil records? Because if a certain part of a sediment core at some time interval is affected by reductive diagenesis, so it will reflect in the bulk magnetic record. So that can be misinterpreted with some, let's say, some climatic process or some other, paleoenvironmental phenomena. So yeah, that means first is the on the quantitative scale, this effects or the impact needs to be quantified. So in the second part now, the oxygen minimum zone and magnetofossils and the relationship with the OMZ, and some of part of my PhD work from a sediment core that is collected from the oxygen minimum zone of Bay of Bengal. So what are OMZs? OMZs are like a persistent layers in a ocean or in an oceanic water column, where we have a reduced oxygen concentrations. So this is a profile. This is a profile from a central region of the Arabian Sea. And before we go to this profile, first is this diagram shows the oxygen concentration at a water depth of 300 meter across the globe in the oceans. As we can see that most of the OMZs are located on the western sides of the continents, like, and the dominant OMZs are like in the Pacific Ocean along this tropical region, in some parts of the Atlantic Ocean, and dominantly in the northern part of the Indian Ocean. Yeah, so this is the part I'll be focusing later on. And this is a profile from Arabian Sea. But the question is, how does these OMGs form? Basically, we have a lot of influx of organic carbon from the continent through the riverine sediments. So when these organic carbon, they sink or they freely fall from the ocean water column, microbial processes starts to degrade it. But within a certain part, let's say 200 meter of the water column, there is always exchange of oxygen with the atmosphere. So to oxidize this organic matter, oxygen is required. But that is replenished in this 200 meter, because it's always in exchange with the atmosphere. But as that organic matter goes down, in the part from 200 to 1000 meter, uh, because of the degradation of this organic matter, the concentration of oxygen gets significantly lowered. And these are the regions, basically, we call it as the oxygen minimum zone. And below 1000 meter, there is most of the organic matter is consumed within the 0 to 1000 meter water depth. So the organic matter will be there, but it will not get oxidized. So that the oxygen demand is very less in this region. So whatever the concentration of oxygen for this region will be the oxygen concentration of top layer minus whatever that is consumed, the oxygen consumed by the microbial processes. So that will be the concentration over here, because this water might be upwelling somewhere else. So yeah, so again, the oxygen, there is a replenishment of the oxygen somewhere else. So because MTBs or the magnetofossils records are, MTBs are sensitive to the redox changes, specifically to the oxygen concentration. So the bigger question here is, can the variations in these magnetofossil abundances, both conventional and giant, can be used to reconstruct the past changes in the marine oxygenation. And another question on top of this is, can magnetofossils serve as a reliable proxies for tracing evolution and intensity of this OMG through geological time? Because it's already, there are several records that shows that the oxygen minimum zones in different parts of the oceans have expanded or reduced over the geological time scale. And why this is important to study OMG? I mean, I will explain in the later slides. So yeah, so for this, we analyzed a magnetic record of a sediment core that we collected in the Bay of Bengal, and the water depth, the sediment core was collected at water depth of 325 meters, which is the present day oxygen minimum zone in the Bay of Bengal. The core length is 2.83 meters, and that score spans last 42 kilo years. And for this site, the majority of the sediments are originated from the Deccan basalts for this region, and locally from the Archean Proterozoic gneissic complex rocks. And this site, the core site, is located at a region where we have a surface oceanic current, and these currents change their direction with the changes in the monsoon. During summer monsoon, the wind flows from this part of the region to, like, from southwest to the northeast part, so it will affect the product, it will affect the direction of the surface current and eventually the marine productivity, because with these currents there will be an upwelling of nutrients, and yeah, so because of the nutrient it will affect the biological productivity. Similarly, during winter monsoon, the direction of the current changes oppositely. So this is a bulk magnetic and geochemical records for this sediment core. As we see that for the Holocene, for the Holocene part and the last glacial maximum, we see a stronger magnetic signal. That is, we have a higher magnetic susceptibility, and yeah, and consistently the S ratio, or the magnetic mineralogy indicator, shows values around 0.9 that could suggest that most of the minerals are ferromagnetic. And in the bottom part of the sediment core, there is a reduction in the magnetic signal, and also the S ratio is also suddenly getting decreased. And yeah, so this part of the sediment core is affected by the reductive diagenetic processes. And the organic carbon record, it shows a continuous increase down core, and it is highest in the part where the sediment, there are probably present day sulfate within transition zone over here. And the calcium carbonate percentage, during late glacial time, we see high concentration of calcium carbonate. That could be probably because of the increase in the marine productivity. And so these changes, the changes in magnetic mineral records, we have analyzed that these are closely because of the changes in the monsoonal intensity. And it is well known that for the last 40 kilo years, the monsoon has changed significantly. For the Holocene time period, the summer monsoon intensity has increased. So yeah, that will affect the sedimentation in the region. And similarly, during so winter monsoon time, there are changes in the monsoon. So this record will come in later part of the slide again. So sorry. So in this profile, the stars represent the layers we selected for the transmission electron microscopy analysis. So yeah, so this sediment core record is dominated by the presence of both conventional and giant magnetofossils. As you can see, a nice picture of giant bullets over here. Also the needle, a bundle of a needle, some conventional magnetofossil with bullet shapes, a tiny needle, and also a short chain of a conventional magnetofossil. To confirm their composition, we did selected area diffraction scanning, also some EDS spectra. So that provided the composition of these crystals. These are magnetite crystals. So again, we did fork analysis for diagrams on selected sample intervals, and these show a dominant presence of a central reach that gives an indication of non-interacting single domain particles. On top of that, we have signatures of detrital minerals through the pseudo single domain or multi-domain signatures in the fork diagram. So, so these are all bulk magnetic records, but in order to link the specific magnetofossil or specific type of magnetofossil abundance to the changes in the OMG, we need to separate out the signatures of these different types of magnetofossil. Not only the magnetofossil, but detrital magnetic minerals also. So before moving to the next part, why it is important to study these OMGs in the northern Indian Ocean. So here, the OMG is it's dominant in both the oceans, like the Arabian Sea and Bay of Bengal, but they are quite different. And actually, how this is, I mean, this is related to the presence of EMTB. I'll explain you. In the Arabian Sea, I mean, literally, we can call it as OMG because uh, it's functional anoxia in that region. I mean, uh, the the depletion of oxygen is of that level that the higher organisms that require oxygen will not survive in this region, and the, the 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 processes that control the OMG in this Arabian Sea are totally different than the Bay of Bengal, mostly because during the summer monsoon period there is a increase in marine productivity over this region, and that because of higher increase in demand of oxygen, there is a consistent presence of these oxygen minimum zones in the Arabian Sea throughout the season. But however, in Bay of Bengal, as we see that most of the rivers in the Indian subcontinent flows into the Bay of Bengal compared to the Arabian Sea, and so this huge flux of fresh water into the Bay of Bengal creates a strong stratification in the water column. So that will not allow vertical mixing of these oxygen rich, oxygen poor waters. So that will hamper the overall productivity. And another reason is the presence of cyclonic situations that also introduces water oxygen into the water column. So strictly speaking, the oxygen minimum zone in the Bay of Bengal, it's not really oxygen minimum zone. The conditions are mostly suboxic in that region, and yeah, you know that the suboxic conditions are, I mean, those are the conditions which are required for the MTBs to grow in that region. And uh, we do some profiles in that region. The yellow dot represent the core location, which is the sediment core is presently located within the present day OMG of Bay of Bengal. And another important thing is when oxygen is not available for the microbial degradation of organic matter, these microbiota shifts to the oxygen that is bound to the nitrogen, that is nitrite or nitrates. So in a strong OMG conditions like in Arabian Sea, there is a denitrification process that is continuously going on. So your nitrite is converted into nitrogen, and that nitrogen, when released in the water column, it oxidizes to nitrous oxide, which is a potent greenhouse gas. So most of the whatever the nitrogen coming through the denitrification process of the ocean, 40% of these is coming from the Arabian Sea, and this denitrification process, which is recognized as a evidence for strong OMGs. This process is not occurring in the Bay of Bengal region. So researchers thinks that Bay of Bengal is on the tipping point. If there is a global warming, so that will acidify the oceans. So that will reduce the oxygen solubility in this region. So this community eventually will shift on the oxygen that is bound to this nitrogen. So if this process dominates in Bay of Bengal, so I mean it will completely change the nitrogen budget of the oceans. So yeah, so these are some of the reasons why we should study the OMGs and how they have varied over geological time scale. So to separate out and link the magnetofossil signatures, we conducted for PCA analysis. And for this, we excluded the bottom part of the sediment core because it is affected by the reductive diagenesis. And from this, we found three N members to 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 represent this for PCA model. And these are the horizontal coercive profile across the central ridge. So EM1, that is N member 1, is mostly of detrital signature. EM2 is detrital plus we have this central ridge signature but of low coercivity. So we assign this to the biogenic soft component. And the last one is detrital plus biogenic hard component. So you can notice that it is difficult to separate out the detrital signals from all these three end members. Maybe probably we need to increase more folk diagrams in this model to separate out these signatures. So now I'll try to link these separated end members and how these are varied over the times or the downcore record. Sorry. So yeah, again the downcore variation record, as we see that N member 1, that is detrital, it has increased for the late glacial to last glacial maximum time period, but it has a lower proportion during the Holocene. But EM2 is dominant, that is detrital plus biogenic soft is dominant during Holocene as compared to the late glacial and uh, last glacial maximum. And for the EM3, detrital plus biogenic hard, it is the proportion is dominating for late glacial and LGM time compared to the Holocene time period. And so I also plotted this overall sea level curve for the last 30 kilo years. As we see that during Holocene, the sea level has increased almost from like for the 100 meters for that region. So to explain this better, I'll explain in two cases. One is where we have a high sea level stand, and another is where we have a low sea level stand, and how this is affecting the magnetofossil and detrital magnetic mineral records. So in case one, during Holocene, as we know that the summer monsoon has intensified, and this is the present day sea level, and the red line is a oxygen minimum zone upper boundary for the Holocene time period. Because of the increase in intensification of summer monsoon, we have a higher sedimentation from both the local river and from these KG rivers, which mostly flows through Deccan basalt. So they will bring, I mean, we can expect that they will bring more of a magnetite, atonomagnetite rich flux. And during this time period, Holocene, we see that the proportion of EM1 and EM2, that is detrital magnetic minerals plus biogenic soft magnetic minerals, has increased. So, so the proportion of biogenic soft magnetic minerals increased. So this happens when we have a introduction of oxygen into that oxygen minimum zone. That means there is a OMG weakening in that region. That's why we are getting a more proportion of equant magnetofossil that I showed previously on one of the slide. And in the second case, during the last glacial maxima, the sea levels were almost 100 meters lower compared to the present day. So we have a low sea level stand, but we have a higher sedimentation. Why? Because during this time period, the intensity of winter monsoon has increased over this region. But the contribution from this river is decreased because of the relative lowering of sea level and a relative change in the depot center of these sediments. That means we have more localized inputs from this river system. That's why we see higher proportion of EM1 and EM3. And EM3 is detritus plus biogenic hard minerals. That includes the elongated conventional magnetofossil plus the biogenic needles that is also incorporated to EM3. So what this represent is that uh, when the concentration of these elongated magnetofossil increase, that means we have OMs strengthening because of the lower oxygen levels. So I mean there are several other records from that region also, and just a sediment core just down to the sediment core which I studied here. That also shows based on these bulk magnetic parameter which has a chi-R and bias-RM very high values, that gives an indication that we have a presence of a non-interacting single domain magnetic particles over this region also. And this study, it's not only based on the magnetic mineral concentration, but they also did geochemical and some paleontological analysis through this multi-proxy approach. They recommend that the OMG, OMG, the strengthening and weakening has happened over last 40 or 50 kilo years. So what are the future research avenues for which I believe from this study are we need to have a combined magnetic and microbiological investigation in search of giant magnetofossil organisms. I believe that is the major step now because investigating water column and surface sediment records like from the Bay of Bengal region might help us to find these giant magnetofossil or giant magnetofossil organisms. Uh, and the next future research avenue would be to look for the to delineate the geochemical conditions that facilitate the growth of these conventional as well as giant magnetofossil in those region, because that will give us a better understanding of the ecological niche and the environmental conditions in that region. So acknowledgement, I would like to thank some our director of CSIR Goa, the project or the ship cell personnel that were involved during collection of the sediment core, some of the people from NIO, also to the Smithsonian Institution for the Doctoral Fellowship, Ken, Tim, and all our collaborators. Thank you.